Boa tarde, Kathrin. Bom dia, apresentação da Dr. Kathrin Nägele. Como está sendo em português de ano, nos 26 anos está contribuindo ao mundo. Então, com ele, hobby inovativo, tem parte de DNA antigo. DNA antigo é algo que está muito recente, provavelmente na área de Caribe. 20 anos passados, mais ou menos 20 anos passados, vai uma conferência na Flórida, com uma parte dos colegas, onde os colegas estão a usar. Nosso país está sendo engenharia na Aruba por muito tempo, para nós vai cultivar aqui informações para a região do Caribe, a região tropical. Sabe que passou por condições de tempo, vamos dizer, de temperatura, que é muito difícil para extrair informação de DNA antigo aqui. Seu colega não logra, porque é um tipo de tal com ele muito inovativo, ele aplica método novo, que ele aplica no Pacífico, ele aplica DNA antigo aqui, e essa que ele aplica na Caribe e também na Aruba. Então, estou muito contente de começar a discutir colaboração com ele, assim, científico, assim, assim, inovativo, e também com o resultado de nós mais compreensão de nós, vamos dizer, de nossa opinião do território. So, Kathrin, please. Go ahead, you have the floor.
Vielen Dank, Harold. Vielen Dank for being. That's unfortunately already where my topic ends. But I'm very glad that you are willing to accommodate me in English this time. To learn about how I, at the Max-Planck-Indstitute Anthropology, study human history. What is human history, or what do I understand as human history? So, today, when we look around in the world, we see a plethora of different cultures, of different languages spoken, different foods that are consumed, different religions that are lived. And what I study is events of the past that have led to the diversity that we see today. So I want to understand, looking into the past, how did humans migrate, how did humans mix, so to build the diversity that we see today. And you can study human history with different lines of evidence. Like, for example, with history, that most of us we know from history lessons in school, where we look at scriptures or letters of things that other people in the past have produced, telling us about the reality of the past. But in my conception, I also include oral traditions that are passed down from one generation to the next, through songs, for example, or poems, because I think that both of these can be equally reliable. And a source of information about the past. And then with languages, we can also study interaction in the past. And I think Aruba is probably the best example with into having so many different influences from the different, also human influences that you've had in the past. And also in other places in the world, like for example the Amazon or in the Pacific, we can see how languages that are more closely related, that are easier, where people have an easier time understanding each other, and languages where people have a more difficult time understanding each other. Sometimes we have languages where we have one language, but then some words are from another language. Like, for example, in the south of Germany, where I'm from, we speak German. We speak a German dialect that is difficult for people to understand, but overall it's German. But we have many French words, because after World War II, we had a French occupation making sure that we would not start Nazi marches again. And also with archaeology, of course, and this is the best place to learn about the archaeology of Aruba. We can identify through similarities in material culture, like for example through the designs in pots, or through similarities in burial practice. We can study connections of humans in the past. Now my field is genetics, and this we can also use to study history. But I will explain more about how I do this. First, I want to say that I don't think that we can rely on only one of those disciplines alone, because each of these disciplines has their limitations. Like for example, in history, those texts, they're often very much biased by the view of the person who wrote them. And this, in the past, and also in the present, was often a European, white, and male perspective. And so we know that people see the world differently depending where they come from, so we need to pull this into question if what, for example, the colonizing chroniclers wrote about the Caribbean when they arrived here is actually what was also the reality, or if maybe they interpreted what they saw through their colonial lens. And also with linguistics, sometimes languages can actually replaced. And the same also with archaeology, where sometimes we have very similar burial practices or very similar ceramics produced in different parts that are not necessarily connected to group history. But when we put all of these evidence together, we can get a much better understanding of the real shape of the human past. But since my field is rooted in genetics, I want to give you a little bit of an introduction today, because I think that this is probably what most people do not have a very close interaction with on a daily basis. So I want to start very simple and explaining what is DNA. So DNA is something that each one of us has, not just each one of us, but every living being on this planet has DNA. Whether it be humans, or birds, or plants, or fungi, or even bacteria, we all have the same language when it comes to our DNA. And we find this DNA in our cells. And to illustrate what a cell is, I've put this shark here because on the shark skin we have these individual teeth or scales. And just like the shark's skin is built of multiple scales, we are built of multiple cells. And in each of those cells we have, again, different organs. Just like we have a heart and a lung and a gut, also our cells have different organs with different, sorry, I'm missing the English word here, uses. So they do different things. One of these organelles is the core of the cell, and the use of this core is to protect the DNA. So inside of this we will find the DNA. And this is a very, very long cord, let's say, that has chemical structures that you could translate into letters. So this long cord holds the information, and it's inscribed not in 26 letters like in our alphabet, but in only 4 letters that have different successions, and so in these different successions they produce different words. And we as geneticists have learned how to read these sequences. They're not in the cell like a long spaghetti just thrown in there, but they're very tightly packed in what we call chromosomes. So these are 23 tightly packed chromosomes, that each one of them consists of a very, very long of those strands tightly kind of curled up. And we inherit half of these chromosomes from our mother, and the other half we inherit from our father. So in our DNA, we have information from both of our parents in the cell. And this is because our parents are mixtures of their parents. Also, they have 2 of these copies, of one from their mother and one from their father, and pass them on to us. But this does not happen in the simple way they illustrated here, where it would assume that this child here got one part of the information from the maternal grandmother and the other part of the information from the paternal grandfather, but instead this is mixed in our parents. These 2 informations from their parents are mixed and then passed on to us in a mixed way. So we are actually mixtures of our grandparents, who are in turn mixtures of their parents and so on. There is one pair of chromosomes that I want to point out that we call the sex chromosomes, because it's the only example where we do not have 2 of the same copies, but there we have 2 different types of sex chromosomes. We have a chromosome, the larger one, that is an X, and when you inherit an X chromosome from your mother and an X chromosome from your father, then this is a person that we would identify as female, then we would address as female. And when you inherit an X from your mother and a Y from your father, then this is a person that we would address as a male. On the genetic level, it's a little bit more complicated because there's deviations from this binary X, 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 Y. We also have people that have only one X chromosome or 2 X and Y chromosomes, so there's a little bit of variation there as well. But the important thing is that the Y chromosome, in any case, is only ever transferred from the father to the son, and then from the son to their sons, and so on and so forth. So with the Y chromosome, we can trace the paternal lineage of a family. There's also correspondence in the maternal lineage, but this is not encoded in our nuclear DNA that is stored in our core, but this DNA is stored in a different organelle that we have, which is called the mitochondrion. The mitochondrion is a very important organelle because this is the organ that produces the energy that our cells need for our daily functions, to walk, to breathe, to dance. So it's a very important organelle, and this organelle comes with its own DNA, which is much smaller than this huge nuclear DNA that we have, and only has one small ring. But this organelle is passed on from mother to her children through the egg cell that she produces. And so because the mother passes on to their children, but then only her daughters can pass it on to their children, and so on and so forth, we can trace the maternal lineage with this mitochondrial DNA. So when we look at how this is then passed through many generations, we have here the example of this is us, a combination of our parents, and now we need a partner who is a combination of their parents. So when we produce a child, this child is actually the combination of his 4 grandparents, and his 8 great-grandparents, and 16 great-great-grandparents, and so on and so forth. And when we put numbers to this, then we can see how fast the number of ancestors that we have explodes. So a child today has 2 parents, 4 grandparents, 8 great-grandparents, 16 great-great-grandparents, and so on. So our 4 times great-grandparents are only 64 people. And when we go back to the time of the European invasion here in this region, then we already have 65,000 ancestors. So with the genome of only one person, we can understand the genetic profile of this individual, but also make assumptions about all of their ancestors. This is not universally true. We can make an assumption about overall the ancestors, but when we try to link them directly, this does not work as well. So imagine a person in Aruba, 1500s. Europeans are arriving and they're having children. So now we have an individual that is half indigenous Aruban, half European. Another region in the world where we find ancestry in Aruba is, for example, Africa, through the transatlantic slave trade. Also, Africans contributed to the genetics of this island. So now we have an individual that is half African and half or quarter European and quarter indigenous Aruban. Another region that has influenced the genetics of this island is other places in the Americas. So indigenous peoples from other parts in the Americas have also either been displaced or migrated to Aruba and also contributed to this. So now we have an individual that is half indigenous South American and then has much smaller ancestry contributions from the other ancestries that were previously mixed into. And when you continue this further and further, you see that some of the components, they become smaller and smaller and smaller. And now I said earlier that before the information is passed on to the child, the information of your parents is mixed in you. But this does not always happen 50-50, but sometimes also 49-51. And because of this, sometimes even though so slightly uneven passing on of information, it can happen that through these generations, you lose the genetic information of one ancestor completely. And so it is difficult to directly link a person today to people in the distant past because this information can randomly get lost. In a population, you might then find one person is linked to it, and their brother is not, because they happen to have not gotten this passed on. So we cannot connect the individual people to the distant past, and this is where ancient DNA comes into the play. Because with sampling the humans in the archaeological record, we can open the window into the past a little bit wider and go back generations from there to understand what happened in the so and so many generations before that, just providing us a much better understanding of the DNA past. And what makes DNA ancient is that, so our DNA is a very, very long molecule, very big strand. In this time, after a person dies and when they are in the ground for a very long time, we have reactions, chemical reactions that lead to a fragmentation of the DNA. So the DNA is chopped into smaller pieces through chemical reactions. And here in the tropical regions, this happens much faster because we have higher humidity than in other places, and these are reactions that are enabled by the presence of water, and then the heat speeds up those chemical reactions. So because of this kind of chopping up of the DNA, one of the properties of ancient DNA is that we have very small fragments that we look at instead of large fragments like we have with modern DNA. Another chemical damage that happens is that one of the letters that we have is changed into a different letter. So it's kind of like a typo in the book. And then we don't know, okay, is that word now actually spelled that way, or is that just a typo? And this makes things for us much more difficult to analyze, but it's also a good thing. And kind of when we represent this in a graph, we call this graph smiley. So here we have more damage at the end of the reeds because they're more prone to the attack of these chemical alterations, and then less towards the center. And we call it smiley book because on one hand it looks like a smiley, but then on the other hand it makes us smile because we can be sure that what we have is ancient DNA. And we can find ancient DNA in many different materials, like for example ancient feathers or shells, cultural artifacts that we can scrape off whatever was in there and try to identify with DNA. Perhaps the foods that were stored. We can extract DNA from seeds and herbarium specimens, and also from fecal matter and dental plaque. Dental plaque is what the dentist removes from your teeth when you're at the dentist. And in the past, they didn't visit the dentist too often, and so foodstuffs or also microbes, or kind of the bacteria that live in your mouth and that regulate your mouth function, are trapped in this calculus. And this can provide us information, for example, of the health of the oral cavity of people in the past, or also when you, for example, have a cough and you cough something up, then this can also get trapped. So it can also provide us about information of diseases that people have in the past. My favorite elements to work with are skeletal elements, and there I have two specific ones that I favor. One are teeth, and the other one is a petrous bone. So teeth, I think we all know. They have the enamel on top, and this protects the tooth, and with it the DNA is stored inside it. The other bone, you will have probably difficulty locating where this bone could be in your body. This is the bone that holds your inner ear. So when you put something in your ear, you will end up in this bone. And this bone is one of the densest bones in the human body. And 10 years ago, someone realized, oh, this also has a lot of DNA, even after a long time, and even in challenging regions like the Caribbean. So this was one of the first steps towards being able to do this work here in this place, because before most of this was in Europe. We then take these elements into the clean room. So this is a room that is separated from the outside with high pressure and is air conditioned, and where we have to dress up in a special way. And I only have this one photo of myself because usually I don't take pictures of myself in the clean room, but I wanted to commemorate this date because this was 6 days before my son was born, and this was the day when I realized I'm too pregnant to continue working in the lab. I could barely close my overall, and you cannot see it, but I'm also wearing rubber boots, and I have to kind of kick off my slippers and then with both legs at the same time go into my rubber boots, and I just couldn't do it that day. So I realized, okay, this is over now. My last day in the lab. We also wear a mask and 3 pairs of gloves, a hairnet, and safety goggles, so that we do not shed our own DNA onto the sample. There's nothing that is dangerous about the sample itself, because even if those humans were infected with something, also the DNA of the bacteria or the pathogens that plague them will be very, very small, so it cannot harm us. But I could harm the success of my studies by just introducing my own DNA through my hairs, through my skin scales, through my lashes or tears. And I want to avoid this because I want to study the human in front of me and not my own DNA, because I think that's pretty boring. We then use a normal dentist drill to drill a tiny hole into the bone and produce this bone powder that you can see here, which is a very, very fine bone powder. and is small enough for us to add more chemicals that then break open also these small bone cells and release the DNA out of those, giving us a nice cocktail of different DNAs. And this cocktail contains hopefully the DNA of the individual that we want to study, but also other DNAs from other sources, like for example from the soil that the person was buried in. There's microbes living in that soil, maybe fungi growing, there were maybe plants that left their DNA, or maybe animals that kind of did their thing there and then also left DNA. And so studying all of this could potentially be very interesting because one could get an idea of also the environment that the individual was buried in, but it's also very expensive because especially in tropical regions where DNA gets smaller and smaller so fast, we have often 1% or less of this cocktail that is the DNA that we actually want to study. And the rest are things that are just also there but are not that interesting to us. And if we now invested time and money into analyzing all of those fragments, this would be very expensive. So we developed also another method where we can fish specifically for the DNA that we are interested in, so the human DNA. And we do this by using a magnetic bead to which DNA sequences are attached that are very similar to the ones that we want to fish for. And because of that, they have a tendency to bind to each other. So we can bind the DNA that we're interested in to this bead, wash out all the rest, and just multiply this DNA, because we have to multiply it so that our machines that read these sequences for us have enough to look at. These are sequencers, and they can translate these chemical structures into letters that are readable for us. And this is what we then call reads, because now, instead of having to look at chemical structures, we can read the sequence, and they don't make much sense to us now, because they're also in fragments and we don't know which word goes where. And for this, to solve this puzzle of which word goes where, what we do is similar to what I do when I have to solve a very difficult puzzle with my children. I use the picture on the box as a reference, right? And this picture on the box for us in genetics is the reference genome, which is a genome that is compiled from different humans from different places on the world, and kind of identifying which is the most common letter in each and every single one of those positions. And then we can give this to the computer and tell the computer, okay, now find the best possible place for each and every single one of those smaller fragments in the reference genome. And this is something that takes a lot of computational resources and time. So aside from identifying a good bone that we can use, which is the Petrus bone, and this targeted enrichment, so when we fish for the human genomes, this is the next ingredients basically that we need in our recipe for success in recovering ancient DNA is very strong computers that have the capacity to do these very big calculations for us. Once the computer is done, it produces what we call a consensus sequence. So it reads basically from the first fragment every letter to the last and shows us then what is the result. And in 99.9% of the cases, the letter will be exactly the same as the one in the reference. And then there's a few positions in the genome where humans actually differ from each other. And these we call these mutations, like for example here in the reference you have a G, but then in this individual has in this position an A. This is a mutation that we call SNP, or single nucleotide polymorphism. And humans are mostly the same, but then some people in some places have different states of this, and this is what we can use. So even though we're so similar, we try to focus on just the positions that are different between humans and concentrate on those to be able to distinguish the populations. But this then makes it seem like it's very easy to separate humans into different protagonists, when in reality it's not. It's just possible because we focus on that. 99.9%, even more than that, we're the same, everyone on this planet. And so with these methods, once we have produced the consensus sequence, we can use it to compare to other people living today and other people living in the past to understand something about the human past. And ancient DNA is now a field that is more or less 30 years old since the first extraction of ancient DNA, and since then has provided us with some new insights, but also resolved some long-standing archaeological questions. And I just want to give 3 examples to show you what DNA can do for us. The first one would be the discovery of a completely new human. This is the Denisovan, a close cousin in our family tree that was identified through DNA from this tiny finger bone here. And this was done by my PhD supervisor, who at the time worked on Neanderthals, which are another close cousin in our family tree. And we already know that humans and Neanderthals inhabited the world at the same time, that we even mixed, and every one of us today carries about 1 to 2% of Neanderthal DNA. But the Denisovan was a human that was not known before, that we did not have a skeleton of. So the Neanderthal was discovered in the Neanderthal in Heidelberg, in Germany, which is very close to where I was born. And so we had a skeleton. We knew before we had DNA that there was another human around, but for the Denisovan, the whole skeleton was never found. It was not found to this day. But through DNA extracted from this tiny finger bone, my supervisor could see that this was not a Neanderthal, but it was also not a human. It was a completely new human species. And ever since, this has informed basically archaeologists to look into where else could we find Denisovan skeletons. Because also Denisovans mixed with humans, and everyone in Papua New Guinea and Australia today, and also in parts of Indonesia, has substantial, up to 5% ancestry that comes from the Denisovans. And so they knew, okay, the mixture must have happened somewhere before humans expanded to here, and we're looking on the Southeast Asian mainland, and have now found more pieces of Denisovan teeth. Still not a whole skeleton, but parts of skulls that now confirm that yes, indeed, there was a different species around that we would not have known about had it not been for ancient DNA from this time of finger bone. In Europe, people that died at the time when we see a transition from hunting and gathering to agriculture showed that these technologies of specific crops and how to plant them, and also ceramics to store them, was introduced with humans from the fertile percent around here, and then made its way all the way up into Europe. And before, this was a big question in archaeology, where archaeologists were fighting whether this was a cultural diffusion, which means that someone adopted agriculture and then told their neighbor, look, I'm doing agriculture now, this works really good for me, maybe you should try it too. This person does it and realizes, yeah, that's really great, I'm going to recommend it to my neighbor, and so on and so forth, and eventually it ends up in Europe. Or if it was humans that started migrating from the fertile percent towards Europe, bringing these technologies. And with DNA, we could show that this was the case, that it was humans who had come. So with this complete transition in kind of subsistence, we also see a complete transition in the genetic makeup of the region. And now something that we can also do is we can recover DNA from bacteria in the past. So to understand what diseases plagued the humans in the past. For example, this is the bacterium Yersinia pestis, and this was identified as indeed being the causative agent of the Black Death that devastated Europe in the 1300s. And now, these very prominent examples, it very much impressed me when I was still a marine biologist, thinking that I would want to study fish and squids for the rest of my life. But then I realized that these are very interesting insights that we can get, but sadly, there was nothing from the tropical regions. And by kind of using the methods that I described before, but then altering them to fit the tropical regions, I started working in the Pacific, as Harold has said before, but I then also shifted my focus to the Caribbean. And so, because we're in the Caribbean, I want to tell you a little bit more about the genetic history of this place, or what we've learned through genetics about the Caribbean. And before I talk about the genetics, I want to give a brief introduction into the archaeology of the Caribbean, just so that everyone's on the same page. What we know from archaeology about the Caribbean is that this was settled already 7,000 to 5,000 years ago during what we call the Archaic Age. And the first occupational sites we see in the extreme ends of the archipelago both in Trinidad and Cuba, and then later also in other islands. We believe that the people back in the day practiced like a semi-nomadic fish hunter-gatherer lifestyle, that and also had some cultural variation because we see sites that feature lithic tools, so tools made of stone, and other that use marine resources like shells or fish bones as tools or as ornaments. So we see a bit of a cultural variation. But one of the big questions is also where did the people come from, and did they maybe come from different places, seeing that there's cultural diversity. And a couple of different regions around the Caribbean have been discussed as places of origin, like for example South America, Central America, specifically the Yucatan Peninsula, because it's not so far from Cuba, and the same is true also for Florida. And now more recently, Nicaragua has been discussed as a point of origin because at the time the sea levels were much lower, and so between Nicaragua and the Greater Antilles, lots of small islands arose that are called the Nicaraguan Rise, and they could have provided steps into the Caribbean. Then, 2,800 years ago, we see a change in the archaeological records starting from the south, and we see an expansion into the islands that is in the archaeological records represented by ornated pottery. So we see that also ceramics are present in the Archaic Age, but now they're very colorful and they have very neat designs. And they make their way to the Greater Antilles. Another thing that is reconstructed for them, based on the pottery styles, is their origin in Venezuela, in the Orinoco River Delta. And when the colonizers arrived, they recorded a language that those people spoke that is most similar to Arawakan. So we believe today that this migration came with the Arawakan languages into the Caribbean. One thing that, even though this was pretty well established, that these ceramics were found in the south, so kind of the northeastern South American coast is very well accepted. But scientists like to fight. It's also here, they made up two different hypotheses on how people could advance into the islands. The first one is the southward expansion hypothesis, where they believe that people migrated first to Puerto Rico and then expanded southward into the Lesser Antilles and also northward into the Greater Antilles. And the opposing hypothesis was the stepping stone model, where the idea is that these migrations of Arawakan-speaking potter-making agriculturalists was using the Lesser Antilles as stepping stones in their advance. And so from the archaeological knowledge, we still had a couple of open questions, like for example, where did the people in the Archaic Age come from, and are they different from the people that came in the Ceramic Age, or is this kind of advance also just a cultural one? So is it people teaching each other, their neighbor, to produce these potteries, or is it with other humans that come from the south? And then also, of course, was it the southward expansion, or was it a stepping stone hypothesis? And to answer these questions, myself and also another team in the US started looking into ancient DNA from the Caribbean, and we successfully retrieved from many different places data. And I just want to summarize today the results we had for these from these genomes of these ancestors. So the first question, of course, was, what does the Archaic Age look like, and is it different from the Ceramic Age? And for this, I want to introduce you to something that you will see a lot in population genetic talks or studies. This is called principal component analysis. It sounds more complicated than it is. I mean, the math behind it is very complicated, don't get me wrong, but what we get out, the output that we get is very simple to understand. So every single dot or star in this case that you see here represents one individual, the genome of one individual. And the closer two dots or stars are, the more closely related the genomes, or the more similar the genomes of those two people are. So when you find a tight cluster like this, you can say, well, these people are genetically very similar and they're slightly different here. but overall I would consider this one genetic unit. So we saw, okay, during the Archaic Age in Cuba and in the Dominican Republic, we see a difference only between Cuba and the Dominican Republic, but not within Cuba, that would explain the different material cultures that we see. And now, when we look at what the ceramic age looks like, if this had been a cultural diffusion, so this agriculture spread not with humans, but just word by word and recommendations from one neighbor to the next, we would expect them to fall on this spot or maybe slightly removed, but not too far away. And if this was new people coming from a new place, then we would expect them to fall somewhere completely different. And so when we check out where they fall, you see they're very far apart, confirming that this was indeed a migration from the south, bringing these new potteries and probably also bringing the Arawakan languages into the Caribbean. And so the next idea, of course, was to find out the continental origins, and I have to say that so far we've not been able to identify. This paper was from 2020. I now have 6 more years of searching for the ancestors of the earliest Cubans under my belt, and I still have not been able to identify them. I always joke that this will be the time when I retire, is when I finally found the ancestors of the Cubans. And well, I hope it's not too soon, because I very much like doing what I do, and it's a lot of fun, as you can see. I get to travel to Aruba, for example. But still, so far, no luck, and probably also in the next 2 years there will not be much luck in finding them. I've already shown that the Archaic Age inhabitants and the Ceramic Age inhabitants, they're genetically different. And what was very surprising to me is that we see very little mixture between those two genetic groups, even though we are sure that they have met. And this is very surprising, because usually when humans meet, they also meet, and this is not the case here. So now we're wondering if maybe there was a cultural barrier, or if maybe our sampling is just not good enough. Because something that you have to know about ancient DNA is that with every genome that we find in the past, the whole picture can completely change again. So right now we think they didn't have much contact, but now I'm working on a project with our Cuban colleagues to really focus on this period where we have this transition, the arrival of the Ceramic Age newcomers meeting the Archaic Age people, and hopefully there we will be able to add more detail to this. When we looked at the Ceramic Age, we could see that their genomes are favoring the stepping stone hypothesis as an advance into the islands. And also something that was already known by archaeology is that the Dominican Republic and the Bahamas have very close genetic connections. There we see in the archaeological record trading of different ceramics, so this was an expected result. And just to kind of show you the breadth, that sometimes we have unexpected results, and sometimes they're totally expected and wonderfully aligned with archaeological evidence as well. Very excitingly, we also have genomes from the mainland, from Venezuela, from a site that is called Las Locas, where people were buried 2,000 years, more than 2,000 years ago. And because this was roughly around the time when the Caribbean was settled, and this is the closest ancient genome that we have to the Caribbean, we thought, well, what if they are the ancestors of this migration to the Caribbean? But finding out that they were not, but that they were their own lineage that we had never observed before. And then finally, people today, at least in Puerto Rico and in parts of Cuba, like Colwyn, which is indicated here by the blue circle, they trace part of their ancestry back to these earlier inhabitants in the Caribbean. So in those regions, we see that the indigenous, they are a mixture of European, African, and indigenous ancestry. But the indigenous ancestry that they have is closest to the pre-colonial inhabitants of the islands. In Puerto Rico and parts of Cuba, in all other parts, we also have indigenous ancestry, but this does not trace to the earlier inhabitants, but is more closer to other regions in the Americas, because in the very colonial times, so directly after the colonial invasion, also indigenous people were displaced to the islands from other places in the Americas, And also within the islands, people were displaced, for example, from the Dominican Republic to Cuba or from the Dominican Republic to Aruba. And you can see that there's a dart of data from the ABC Islands, even though they're also part of the Caribbean, and they're a very interesting place to look. So this is why more than 6 years ago, we reached out to the museum here and we partnered up, and I've worked very closely with Harold on providing perspective, a genetic perspective also on the history of Aruba. And here, I'm not an expert in Aruban archaeology. For this you have Harold, for this you have this wonderful museum here. But just to kind of put the sites that we looked at into context, we have in Aruba also an archaic age and a kind of ceramic period, one could say, we call it a little bit differently, and they're slightly shifted from the period that we see in the Caribbean. And the sites that we looked at, one in Colorado, dating to 1,600 years ago, and Kanashito dating to 1,800 years ago, they both fall within the archaic age of the island of Aruba. And then we've also looked at the site, Serenoka, that falls within the Kakatiyo period. So this has lavaculoid ceramics that are not, well, this specific type of ceramics is not found in the earlier periods, but more simpler ceramics are found. And now, of course, we wanted to understand this arrival of these ceramics, does that also come with humans? And where do the archaic age inhabitants come from? So specifically this genetic difference in the Kakatiyo period, also where does this come from? And so I'm going to bother you again with the PCA, the principal component analysis, but now you're already experts. You know that every dot is one genome, and the closer they are, the more closely related they are. So on this genetic map of the archaic age in Cuba and the Dominican Republic and the ceramic age in the Antilles, we're now going to add a couple more groups that we have from the mainland. So these are all ancient genomes from Brazil, Colombia, Peru, and Mexico. They kind of spread along this line, so I would call this kind of the continental part, and this is the island part, and then the archaic slightly moving through. And now we also want to look where Las Locas, which is the next closest, would be falling, falls on this continental part of this plot. And now we can see where we kind of Shito and Colorado, so the archaic age people, and they fall dead on where also Las Locas is. So very similar, genetically, they're most similar to ancient individuals from Las Locas and Venezuela. And when we look at Serenoka, this individual falls in between Kanashito, Colorado, and the ceramic age in the Caribbean. So it shows that there is indeed a genetic difference between this individual from the Kakatiyo period and the preceding archaic age individuals there. But when we summarize this to understand what this means, right, I should also mention that there is already data from Curaçao, and they look quite similar, but not quite the same. They're even more slightly removed towards the ceramic age in the Caribbean, but very similar to Serenoka as well, and they're also from this Kakatiyo period. And now when we think about, okay, what does this now mean in much simpler terms, then one thing that I'm very confident to say is that the earliest inhabitants here from the archaic period arrived 1,000, 3,000 to at least 1,600 years ago. They were probably coming from the mainland, from what today is Venezuela. But what about the Kakatiyo period? Here it's a little bit more difficult because what we see is that Serenoka is a mixture. And one part of this mixture is Las Locas, that is for sure. And the other part of this mixture, I know in the plot it looks like it's the Antilles, but I would now say that this is a much broader ancestry that we do see in the Antilles, but we don't have any representation of what this ancestry would look like on the mainland. And this is why I'm now calling it a little bit more broadly Arawakan ancestry, because we know that those people migrated from northeastern South America into the islands speaking Arawakan languages, and we also have still Arawakan languages on the mainland today. So before, I think that therefore, we have different scenarios on how this shift from Serenoka, from Kanashito could have happened. One is that the Archaic Age came to Aruba, and then there was a second migration from Nägele Antilles into Aruba, producing this mixed individual. But I think it's equally likely at this point, because we cannot say we don't have data, that this comes directly also from the mainland, so we have a second migration from the mainland of this Arawakan ancestry. And now to complicate things even more, there could be a third scenario where this Arawakan ancestry and the Las Locas ancestry mix, but on the mainland, in Venezuela perhaps, and then from there come already mixed into Aruba. So these are three different scenarios that right now I'm not confident to say one is more likely than the other. But for me this is great because it means that I will get to continue to work on this, and I get to continue to work with the archaeologists here, and hopefully also be able to come back to Aruba one day. And well, with this, I'd like to say Masha Danti, but I want to give specific thanks to the museum here for the great collaboration, for Harold to showing me around the island, and especially the sites that we've studied. This has already provided me with a new, very rich insight, and inspired me a lot to do more analysis, to do different analysis, to look into other directions. So this was really, really great. And I also want to thank my group. We're the Tropical Archaeogenomics group, which you can also follow on Instagram. And the work that you've seen today was not analyzed by myself, because I rarely get to do this fun thing anymore, but by my PhD students, Hunit, Eleni, and Beatriz. And with that, thank you for your attention.
Thank you, Kathrin, for that awesome presentation. I'm going to give a bit of a context about the information that we got and how important it is. The part of where people came from, the first archaic people that came from, is a question that we've had and we've discussed for years. Even the guy before me, Alpha State from the 90s, he had the idea that maybe they might have come from the coastal area of Colombia, maybe they might have come from Venezuela, but now we know that they came from Venezuela. So when we see here the map, we see the archaic sites. So Las Locas is a site which is dated 400 BC to 380. It's a site located in Lara, and there they found 25 burials. And these burials were all not only primary burials, but also secondary burials. Secondary burial means that they first buried the person, and then they reburied parts of the bones of the person after a while. Aside from that, they also have corn, and also they have ceramics, polychrome ceramics, but also tetrachrome ceramics. So they had more widely developed ceramics than we think or than we expected. Here in Aruba, we found on all the archaic age sites, we found ceramics on the floor, but I worked in Roy-Briganosa, in excavation there, and there we found ceramics within the archaeological context below the surface, so that means that they did have the ceramics. And the interesting thing is that the ceramics that we find for the archaic are the ceramics that were more related to travel because they were thinner compared to the Capitin ceramics which were used for food stuff and food preparation. So when we look at the dates that we have here, Spanslahun falls within the range of Las Locas. Kanashito also falls within it. Cerro Colorado also, but then we also have Isla that falls within this range. So here we have 4 sites which indicates that it's a possibility, and I think I'm quite confident that all the people that came during these periods were originated from Las Locas in Venezuela. The Isla one is very interesting because there my colleague Raimundo Deikov worked on it, and they found a monkey burial, but it was a secondary burial with a female. And we've been wondering how was that possible, because before we thought it was from the historic period or Castillo, but when we got the carbon date, we found like it's an archaic burial, but also with a monkey, which is also secondary burial. And Aruba is the only place within the whole region where a monkey is buried with a person, and also in a secondary manner. So that's a very special site that we have here. I also want to mention this site in Antiflip. And the flake is considered ceramic period Kakatiosite, dated 980 to 1550. But here we see that we find somebody from the Archaic period within it. So that shows also that the Archaic were much more distributed throughout the island, and also the component of corn that they had, the cultivation of corn, was also something that it seems to me that was more widespread than we think. But then we have these sites which are much older than Las Rocas, so we still have more research to do, because now I wonder, okay, these ones fall within the Las Rocas period, so that they came from Lara, and they did some modeling, seawall modeling, and they would have, so if you see like in Australia here, we have Lara here, Aruba's here, they would have followed the coastline and then crossed over to Aruba. And the interesting thing is that this was something that could be done year-round, but in January was the best time to do it. But also then the other question would be, did the Kakatiyo know that there were people here when they arrived, because they might have come from a different direction. So we have a lot of interesting questions, and thanks to the research of Kathrin also, as I mentioned, she dedicates on the Archaic period of the Caribbean, which is very fantastic, because now we're able to do more research and understand better where the people came from. Okay, thank you for your attention.